Hello, my name is Malay Basu. I'm a faculty at University of Alabama at Birmingham, and this is a poster presentation, and today I'll be talking about the genomic determinants of a disease called thrombotic thrombocytopenic purpura, or commonly known as TTP. Now, TTP is a rare blood disorder, and the disease is known by having blood clot all over the body, and left untreated, the disease is potentially fatal. And it's also one of the characteristics of the disease is that the skin, because of the bleeding, shows purple lesions all over the body, and thus the name purpura came in. Purpura literally means the purple lesions. TTP can be of two types. One is an inherited disorder, and the other one is an acquired adult onset, and this is an autoimmune disorder. In spite of several years of studies, the etiology of the adult onset or the autoimmune variety of TTP is not really known. What is known is that there are two major players. The first major player is an enzyme called ADAMTS13, and this enzyme actually cleaves another protein called VWF. VWF, it mostly acts as a glue, and when ADAMTS13 activity goes down, either because of a inherent deficiency or an autoimmune response against ADAMTS13 protein, then the VWF actually goes scot-free, and then it actually acts like a glue to create clots all over the body. Now, because the other genetic susceptibilities of the TTP are mostly unknown, we actually decided to carry out a whole exome analysis of the TTP patients. TTP is a rare disorder, and finding samples for the TTP patients is a very hard job. We managed to get 40 TTP samples, and we actually matched them with a 15 control. These controls are taken from the same background as the TTP patient. On average, we found around 13,000 non-synonymous mutation per sample. So first thing, we checked whether there are any mutations that are present only in the TTP sample but not in the control populations, and we found there is no such mutations. So TTP is not a single mutation or single gene based disease, and there are probably more than one candidate genes involved in the TTP disease progression. So we decided to create an association study. An association study is mostly done by the statistical test of the prevalence of a particular mutation on the disease versus the control state. We used Pling software to carry out the association test. Particularly for these association studies, we actually used various filtering criteria. One of the most important one is to discard all single nucleotide variants that have higher than 5% prevalence in 1000 genome. Thus, this analysis is entirely based on rare genomic variants. This is a Manhattan plot, and immediately from the plot we can make out there are several candidates. Now, one important candidate that comes out to be one of the top hits is a gene known as ANKRD36C. Virtually nothing is known about this family of gene. This particular gene belongs to the ankyrin domain containing protein family. There are 3 paralogs known in human genome. Here is the result from the association test, and it shows that ANKRD36C as the topmost gene. And when we performed the Geneset enrichment analysis, we found that not unexpectedly, the top pathway hits are from the immune reaction. But what was surprising to us was that there are several pathways related to glycosylation, particularly O-link glycosylation pathways that came up in the hit, which shows us that most probably a glycosylation defect is one of the causative factors for disease progression. We confirmed the ANKRD36 involvement by carrying out a SCAT analysis. This analysis is based on gene, not for individual alleles. We found that ANKRD36 gene really pops up as one of the top hit candidates. Thus, it proves our earlier finding that ANKRD36 as one of the major genes involved in TTP disease progression. We then created an Episteric network. In this case, the Pling software was used to carry out Episteric relationship between alleles, and then we created a network out of these Pling results. In this network, the genes are nodes, and 2 genes are connected by an edge if there is an Episteric relationship between them. The p-value of the relationship was used as a weight for the edges. In this network, we found 3,500 nodes with a clustering coefficient value of 0.0 37, and we used a software called CytoHub to find out the hub genes in this network. And it turns out the ANKRD36, as we found in our earlier analysis, is the topmost hub genes in this network. When you used all the hub genes in this network and carried out a Geneset rendition analysis, we found that our earlier finding that the glycosylation pathway defect may be a major causative cause is actually confirmed. Carried out mass spec analysis of the samples, and here is a hierarchical clustering showing that the TDP and the control samples do indeed differ in their glycosylated protein profiles. We carried out a differential protein expression analysis using the mass spec spectral count data using a R package called MSMS test, and we found there are 42 proteins that are differentially expressed between TDP versus control samples. Interestingly enough, all the overexpressed glycosylated proteins in TDP samples belong to immune and inflammation pathways, and quite unexpectedly we found that the underexpressed glycosylated proteins in TDP samples mostly belong to HDL pathway. This is high density lipoprotein pathway. So our conclusion from the analysis is that we identified the involvement of the ankyrin domain repeat containing proteins in TDP for the first time. We showed that the glycosylation defect was one of the causative factors in TDP disease progression. This glycosylation might actually affect the autoimmune reaction against aromatase 13 proteins. We also found, quite unexpectedly, that the HDL biosynthetic genes are also involved, are also repressed in TDP patients. So this work is in collaboration with Long Zhang, the University of Kansas Medical Center, and his lab mates, particularly Bikram Pillai and Philippi and Ligia from my lab. Work is supported by grants from NIH and UAV Health Science Foundation. Thank you very much, and I'll be happy to answer any questions that you might have.